Yeah, well, it's so nice to meet you. Dr. Chatfield has always said such amazing things about you. And
She's great.
I remember like when we got our diagnosis, she was like, one day you'll have to go to Johns Hopkins and meet Dr. Vernon. And I was like,
Yeah.
what is she talking about? Because it was like, he was a 2-week-old baby and I was just
Yeah, no,
not even
Katie's
2 weeks.
great. We talk all the time. We collaborate on research and we
Yeah,
share a few patients. So yeah, she's
that's
great. Yeah,
awesome.
you've got a good team there. I
Oh
mean,
my God.
we're always happy to see anyone in our clinic, but you've got a great team. So
Oh, she is the best. We are so lucky that we just always feel so fortunate that we're like here and that she's here and that became our cardiologist. So yeah,
yeah.
big fans. So yeah, we'll keep it short because I know time is of the essence, but
Mhm.
I'm just so curious. I would love to know like how you got involved with Barth syndrome research and the clinic and like how that all started. Um, and then maybe if you could talk a little about the TAZPOWER trial and what that was all
Yeah.
like. And yeah, just,
Sure. So
yeah,
I'll just ramble and just interrupt and ask whenever you want, any
sure.
more details. Yeah.
Yeah.
So the way I got into Barth syndrome, I think like anything, it was a little was pretty serendipitous. So when I was finishing my fellowship at Johns Hopkins and looking for jobs, geneticist at the Kennedy Krieger Institute who was sort of nearing-ish retirement, and they wanted to hire someone who could both see patients at Johns Hopkins and also start seeing some of the patients at the Kennedy Krieger Institute for when the prior geneticist, Dr. Richard Kelly, was getting ready to move on or possibly
Mhm.
retire. And he had been very early and instrumental in describing the first patients with Barth syndrome. And so he had followed a number of patients. And overall, when the community heard there was a new physician interested in Barth syndrome and taking care of patients, the foundation immediately supported my role, and we just started seeing, I mean, it was just like a deluge. So we
Yeah.
thought pretty quickly, so this was 12, 13 years ago already, we got to start a clinic for this. And so we established the interdisciplinary clinic, which has been running, I think, 12 or 13 years already. And we've put together a great team. And from there, I thought, well, if these are the patients I'm seeing, then this is really where I should direct my research.
Mhm.
And that's, sort of the short of a very long story.
That's nice that
Yeah.
you like it's nice that you got to continue the work that had already been started.
Hmm.
And that, you found it interesting and wanted to keep going with it.
Yeah.
How does starting a clinic, like what does that mean and what does that entail?
Yeah. So there's a number of parts of it because we always think, as physicians or practitioners, we know what families need and families want, but actually what's the bigger part of it is what the families say they need and want. So we worked with a number of families and asked what kind of practitioners they would want to see if they came to a full day clinic.
Hmm.
And some of the things were fairly obvious. So, hematology, cardiology,
Hmm.
my role in genetics. But then, we recognized that families really wanted to see a dietitian and really wanted to see a physical therapist. So I picked some colleagues who I thought would be really, really wonderful with, first of all, kids, with learning about a rare disease, with taking on a challenge, because at the time there wasn't a birth clinic in the US.
Hmm.
And we got together pretty quickly as a group and went to our leadership at the Kennedy Krieger Institute, who immediately was like, yes, start this clinic. They really didn't ask us to do any like prove we could make money or anything like that. They were just immediately supportive. They said, yes, do it. And so pretty quickly we were able to get this lined up.
Wow. Okay. So
Yeah.
then when people come to the clinic, you
Hmm.
said like it's an all day thing. Yeah. So
Hmm.
can you tell me more about what happens
Yeah.
with that?
Yeah, sure. So we make sure that families are getting what they want from the clinic. So there are some families we have a few families who are local and we see them a few times a year and they don't need to see every specialist.
Hmm.
There's some families who are traveling internationally or traveling from quite far away and they know they're only going to get to us maybe once or once every few years and they want to see everybody from start to finish. And so we actually send out sort of choose your own adventure sheet to families and say, who
Hmm.
would you like to see? What are the services that you're interested in? What are your biggest questions? And then make a schedule based on what families want. So the last clinic we had, the first week in September was full. We saw 7 boys and men with Barth syndrome, which takes up the whole day. But we had one family who only wanted to see hematology. They had everything else covered. A new patient who wanted to see everyone, so they were there the whole day. And so it really depends. We get lab work done, echocardiograms, EKGs. We try to arrange a nice lunch so the families can eat together. So,
That's awesome.
yeah.
Okay, so then tell me about TAZPOWER and about like how that all came about. I feel like we kind of came in at the very tail end of what we thought was, the approval process and things, and then now we've kind of been learning as we go. So it's just interesting to hear about how things started. Yeah.
Yeah, so a long time ago, right after we started the clinic, my lab focuses on cellular models of mitochondrial disease. So basically, we use CRISPR editing, which you may have heard of. It's a hot topic right now, but it's been around for a while, to introduce mutations into the TAZ gene in different cell models. And I had a wonderful grad student. She now works at a gene sequencing company who made a cell model of a known tafazin mutation. And we found all these different mitochondrial changes that were going on in that cell model. And we thought, well, what if we can use some small molecules to reverse some of these changes? So we actually started rooting around in the literature and seeing what's out there, what interacts with cardiolipin, what is a possible, just something we could try. And we hit on, it was at the time, it was, I mean, elamipretide's gone through a lot of names. So at the time, it
Yeah.
was SS31.
Hmm.
And I had another name called Bendavia. And we contacted the company with two questions. One, could we get some of this compound to test in our cell model? And two, would they be interested in Barth syndrome? Because it seems like the compound, the drug, which had been tested in other medical conditions and seemed, safe. It seemed like something very reasonable. And so on both parts, yes, they were interested in both parts. This was actually, I think, a few of the people who are at Stealth now were there then, but it was a different group of people we were talking to for the most part. And
Hmm.
so they both sent us some of the compound, which we were able to test in our cell models and did show that it actually reversed a number of the mitochondrial changes that we had seen. And immediately started talking about a clinical trial. And it was a lot of this was very serendipitous because a lot of the phase 1 work had been done in other disorders.
Hmm.
And so the like how the drug was processed in the body was known, had safety profiles were known. So we had a huge head start. So there was a lot of luck there. And because we already had the Barth Clinic, we'd been tracking natural history for so many years. We actually, at that point, had clinical endpoints we thought we could use. Because there's
Hmm.
two parts when you do a clinical trial. One is having the drug you want to test, but the other part is, well, what are you going to test? What are you going to measure? How are you going to prove something works?
Hmm.
And so we worked with the company, I think I can say their name, Stealth. We worked with the
Yeah.
company and to look through all of our natural history data, all of our publications, and say what would be a reasonable endpoint. And at the
Hmm.
time, and it's still being used as skeletal muscle strength and exercise tolerance. And of course, a lot of secondary endpoints. I learned a lot about clinical trials, like what the FDA needs to see to prove something versus what we're interested in seeing are actually
Yeah.
sometimes a little bit different. Like we wanted to know, was heart function changing? Were lab
Hmm.
parameters changing? But really, there were some very specific things that had to be used as outcome measures.
Hmm.
And so, given the wonderful Barth community, we enrolled that study really quickly within a few months, which is, I've done a number of clinical trials now, so it's really unusual to fully enroll a clinical trial within the speed that we did. And so we were
Yeah.
able to, get that trial going really quickly. So I have to give a shout out to all the men and families who contributed to the study because they didn't know I mean, they knew they were doing a clinical trial, but they didn't know if this was going to work. They didn't know if there were going to be
Hmm.
side effects. They really just took a risk. And so amazing.
Yes.
Yeah.
Yeah, we keep saying to like friends and family that having the access to elamipretide or Forzinity now, but having that and then
I
having
can't
that,
adjust to the new name.
I
I'm going to
know,
call
right?
it I
I know,
can't. I've been
elamipretide
doing,
for life.
yeah, yeah.
But like having that available to Gilbert so quickly after his diagnosis was the work of years and years and years of everyone who came before us. And like you're
Hmm.
saying, like the men and the boys and the families that decided to take a chance and do the trials. And so, when we think about like advocacy work and being involved in the community, we're always just thinking about like how it's kind of like a pay it forward thing. Like,
Yeah,
you know,
yeah,
you everybody who did that years before I'd even heard of Barth syndrome, like this has all been going on. And just how grateful we are to everyone who's, you know, done all of this before because now we're here. And then whatever we're doing now is hopefully what makes it easier in the future for future
exactly,
babies born with Barth and hopefully other mitochondrial conditions like if more research can happen. And yeah,
exactly.
that's really cool. So, um, when you were talking about the endpoints, are there certain ones that like are kind of repeat things? Because there was another family we talked to, and she's on elamipretide through expanded access,
Mhm.
and we were talking about like 6-minute walk tests and things like that. So are there certain like types of endpoints that are commonly used in trials?
Yeah. And the FDA experience has been a long and
Mhm.
sort of roundabout one, and
Yeah.
I've learned a lot. I've learned a lot
Yeah.
about this. I knew nothing about any of this going in, but you actually, in designing a clinical trial, work with the FDA to
Okay.
design endpoints. So we worked with the company, we designed endpoints that we thought were important, but then you go to the FDA and say, here's what we're planning on measuring. Are these acceptable to you
Hmm.
as outcome measures? And then they'll give you guidance about, there are some outcome measures that we offer, and they're like, these are not primary endpoints. These haven't been validated. This isn't an acceptable test. You can use
Okay.
it as a research endpoint, but this is what we ex so 6-minute walk test is has been used to as an endpoint for trials, lots and lots of other measurements as well. And so that's kind of how so it's kind of combining what we know about the disease
Hmm.
and about the natural history of the disease, but also what the FDA accepts as an endpoint. And then
Hmm.
kind of predicting, do we think we can see a difference, which is a whole other part of things because there's a
Yeah.
lot of guesswork up front. You have to take a lot of leaps of faith in the beginning. So,
Yeah.
yeah.
So the one that sounds like is the clinical endpoint that is part of the approval, the knee extensor. What
Mhm.
is how do you measure that? Like what does that look like?
Yeah. So all that testing was done by Brittany Hornby, who is a physical therapist who was with our clinic for more than a decade. She's working somewhere else now. And we miss her. It uses basically a device that measures the amount of pounds that someone can pull or push. And so it gives a
Okay.
number. So the examiner holds the device and then the study participant pulls or pushes, and it basically gives pounds or newtons about how much they're pulling or pushing. And so for the knee extensor, that's basically kind of the muscle that helps your knees extend away from the body. And we know that in Barth syndrome and in most mitochondrial disorders, it's the like the hip muscles and the shoulder muscles that are most affected. It's those big muscle groups.
Hmm.
And in our natural history work for years, we found that almost every patient that we measured with Barth syndrome had abnormal knee extensor strength. So, and flexor strength as well, actually, and as well as in the hips. And so we knew that if we saw improvement, that would be significant because that's not the natural history of Barth syndrome.
Okay.
So it gives you a number and something that you can measure over time. And also, what's really important to me is something that's not invasive. So,
Yeah.
not, there's nothing risky about doing it. So.
Yeah. Yeah. So is it like you're like is it like that thing when you're like in the weight room where you like you like
It
have your
kind
like
of,
you
it's got
push
like a
it?
strap and kind of like a box that measures and you pull. So it's like it's handheld, like we carry it around.
Okay.
Yeah, yeah. Yeah. It's
That's really
portable.
interesting. I know. Well,
Yeah.
people have asked me too. They're like, okay, well, I tried like researching this. Like, what does that mean? I was like, well, I'm gonna
Yeah.
ask because I
Yeah,
don't know either.
it's a device. We've brought it to lots of the Barth meetings when we do some of the natural history
Yeah.
stuff. So anyone who's participated can describe what it looks like too.
Cool.
Yeah.
I'm gonna go find a picture and
Yeah.
put it in. Wow. Okay. So then I guess what are your thoughts on, where do we go from here? I mean, obviously some work to do in terms of the labeling.
Hmm.
Of the drug, but like it's very exciting that we have,
Yeah.
the first approved treatment. And years and years of work has led up to this.
Yeah.
What do you see as kind of like what else is being researched for Barth
Hmm.
syndrome
Yeah.
or mitochondrial diseases, or what are you working on right now? Yeah, what are some other things that are going on?
Yeah, so there's I can split this into two parts. The first part is what we see for elamipretide, which you already kind of got at the uh, some of the more important work to be done there, which is figuring out a way to extend the label indication so that people of all ages can access it, having it tested and approved. We have to do a confirmatory trial, which will
Mhm.
be mostly in Europe. So that study as well. So there's those parts of things, and I think we will learn a lot more about elamipretide and how it affects Barth syndrome, the natural history of Barth syndrome over time. I think we're going to learn a lot of new things
Mhm.
by basically having a register where we would follow all of our patients who are on drug and off drug for an extended period of time. I've been a part of a number of these uh, sort of observational registries over time, and you learn a lot about drugs and
Mhm.
how they act and the things that they can do the longer you watch. So there's that part. And then in terms of other treatments for Barth syndrome, I mean, I envision for any disorder, whether it's rare or common, ideally there would be a toolkit of different therapeutics that are available. And, patients and families can look at what's available and figure out what's best for them. So
Hmm.
right now, elamipretide is the approved drug, but, hopefully in the future there'll be many other different kinds of interventions that are going to be available. And then families can choose, this is good for me, this is good for me, this combination of things is good for
Hmm.
me, kind of the way you would treat, a common condition.
Yeah.
So there are a number of things being worked on. Gene therapy is, something that has been is under investigation in a number of labs. And ultimately, that's really where you repair or replace the gene that isn't fully functional.
Hmm.
So that really gets at the cause of the disease. So there's a lot of exciting things happening in gene therapy. So, keeping an eye out for that. I know that there's a group at Tufts that was looking at an enzyme replacement therapy.
Hmm.
So I think that's an option for another toolkit. There's a recent publication by Jason Moffat's group. We helped a little bit with that, but it was really Jason Moffat's group in Toronto that identified kind of the enzyme that creates monolysyl cardiolipin.
Hmm.
And it's called ABHD18, and there's a possibility that inhibiting that could actually allow sort of the nascent or like original cardiolipin, if you want to think about it, expand. And so that's a potential therapeutic that's out there as well. So there's a number of things that are kind
Yeah.
of in the pipeline. So
Yes, I saw an article about that one, and I was like, I found that very interesting that it's like you go up higher up in the chain of what's going on,
yeah,
and it almost seemed like a double negative or so, because yeah.
yeah. And there is actually a precedent. There's a number of disorders that are treated that way. Like tyrosinemia is one of them, which is a kind of a liver amino acid disease. And there's a small molecule that inhibits the pathway kind of upstream so
Hmm.
that the bad stuff isn't created and there's just kind of good stuff hanging around.
Yeah.
So this would be a similar approach. So, it would be a nice one also. So.
Yeah. Yeah, it's interesting. I was like, okay, so you turn that off and then it actually like works because
Mhm.
you have the mutation.
Yeah, and it's funny with the way cardiolipin is remodeled. There's a number of us who've been, studying cardiolipin for years. I'm like, this pathway is just so bizarre because you have
Hmm.
a nascent cardiolipin that has four chains on it. Then this
Hmm.
phospholipase comes and plucks one off and makes the
Yeah.
monolysyl cardiolipin, and then tafazin has to put one back on. And we all feel like, I don't know, there's got to be some reason for it, but why? Why does
Yes.
this happen? Why can't you just have the nascent cardiolipin? So,
Yeah. Yeah.
we've spent some time looking at that actually. We've some of the lab work we're doing now, and some of the work that we've published is like if you stick abnormal side chains onto cardiolipin, does it change the function? And we're still trying to learn a little bit. Does it actually change anything? We don't know.
Hmm. Yeah, I was asking Lindsay about that, and she was telling me that like the nascent cardiolipin like functions very similarly to the mature. So what you're saying because it
Yeah.
has the four chains. That's very yeah. And my husband's a science teacher, and he did like a lot of reading about
Yeah.
all of this, and he was saying that like the tafazin gene is really old. Like it's in like I don't know, it's like in yeast or something crazy. It's like
Yeah.
millions of years old.
But I think there's a couple of trivia points about tafazin that I love. So the first thing is, did you ever read about how it got its name?
No.
But the
How did
clown, Mr. Tafazzi? Yes, I believe it was an Italian group that was looking at the sort of a like homology of the gene and what it might be structured as. You
Mhm.
have to check me on this one, but I'm pretty sure they were looking at the structure and they realized it didn't look like any other protein they were looking at, and it just looked strange. So they named it Tafazin after this like jokester, this Italian jokester named Mr. Tafazzi. So it's named after this Italian like kind of clown guy. Um, but what's interesting is exon 5 arose between great apes and humans. So the exon 5 of tafazin is only in humans. It's not in any other species, including great apes and all the way down. So it's just kind of,
Interesting.
yeah, it arose really, really late in evolution.
Yeah, yeah.
So there's no disease causing mutations in that exon because it doesn't really do anything. It just kind of hangs out. So
Very interesting.
yeah.
Yeah. And yeah, Andy said something about like in the development of elamipretide to begin with, like it was an accident or something, like someone injected a rat in the wrong place or something like that.
Yeah. And they realized that it interacted with cardiolipin. Yeah, yeah.
Yeah, it's
Yeah.
funny how so many things in medicine and science like are kind of just like happy mistakes, or you happen upon it, something you weren't necessarily looking for, and then you find that, and
Right,
yeah.
like penicillin, right? Wasn't it was
Mhm.
just like a mold
Yeah.
that they figure out. Yeah, you can learn. I always teach my students because they get frustrated when stuff doesn't go right. Of course, I'm like, you can learn a lot from your mistakes. And who
Yes.
knows what you're gonna, not even mistakes, just in science, we call it the lab gremlin. There's just like the gremlin comes in one night and nothing works. I'm like you can learn a lot from the lab gremlin as well.
The lab going.
Yeah.
That's funny. Oh my goodness. Well, yeah, no, this is all very interesting and it is exciting. So for gene therapy, um, would it be like because like my understanding of like my limited understanding is that like each there's like different mutations. So like that's why Barth syndrome can present kind of differently in each individual. So would you have to have like a different type of gene mutation or gene therapy for each mutation?
So there's a couple of approaches to gene therapy. One of them is gene replacement therapy, where you basically put a whole functioning copy of the gene in, and
Mhm.
that's usually administered via like adenovirus. So that's you don't that is mutation agnostic. It doesn't matter what the mutation is because you're just putting the whole functioning gene in.
Mhm.
What there's been a lot of news about is things like CRISPR editing, or which is where you actually change a specific mutation, or ASO therapy, which is where you can kind of inhibit the bad part of a gene being made. Those are mutation specific. And so, if you think about CRISPR editing, where you design a specific, it's like designing a specific recipe to change a specific site,
Mhm.
would be there are many tafazin mutations that would be amenable to that, but each one would require a different CRISPR molecule.
Mhm.
There are
Mhm.
people out there working on CRISPR platforms where it's just sort of like a base platform, and then you use the same platform to just design different mutation corrections. But as far as what I'm aware of for gene therapy in development now, or being studied for Barth syndrome, it's all the complete replacement. So putting a whole functional tafazin back in. So that would work for
Yeah.
conceivably everyone.
Everyone. Okay.
Hmm.
Yeah, because I just remember like when we getting his diagnosis and the geneticist was like, okay, so on this, like this is the specific location, and instead, I can't remember what letter it was. It was like
Yeah.
either instead of a G, it was an A, or it was like the other way around. But
Yeah.
I was like, that's crazy. It's just that one switch. And
Yeah. And we all have millions of changes from our parents, that many. And it's just some of them happen to be in spots where it does something, and some of them happen to be in spots where it doesn't. But we've all got them. So.
yeah, yeah, it's just it's wild. Like it seems like the potential in the next, 20 or so years, like the things that we're gonna figure out are crazy just
Yeah.
because of where we're going with the technology and everything.
When I originally in med school, I remember saying, because I always knew I wanted to do genetics, saying I want to go to residency for genetics. People were like, "There's no therapies. You can't do anything. Why would you pick a field like that?" And I was like, "Well, lo and behold,
Yeah,
first of all, it's just always been exciting, and I've always been interested in genetics. But things are just changing at a breakneck pace. It's amazing."
yeah, they really are. It's wild. Well, wow, that's very exciting. I guess so for my last question, since we're almost out of time, for families, there are some families who listen to this who have other like types of rare diseases, and their um, groups and organizations are at kind of different points along the spectrum. Like when you talk about like natural history and things like that,
Mhm.
what do you, as a researcher and a clinician, like what do you feel like is what are some helpful things that rare disease groups can do to better like get researchers involved or to get to get things off the ground for things like natural history and such.
Yeah. So starting a family group, a place where families can center, can come together, is
Mhm.
really the first most important part. So just so you know who's out there,
Yeah.
because the more people who are involved, the more data you can get. And I do think before there's even a hint of a therapeutic, it is good to start a natural history study.
Mhm.
It's important to start it the right way. And what I mean by that is you want to create a natural history study that's usable, because if it's, I'm trying to think of the best way to say this. There are some things that are able to be used for that the FDA will look at, and some things that aren't. So finding reasonable way to collect the data that's usable. So just talking to other people who have done it, bringing in other groups who have had successes and have done this, and just start collecting that data. The more, the better. The earlier, the better. And again, talking to other groups who have done this, I think there's a lot
Mhm.
of successes out there and a lot of learned experiences. So
Yeah. Yeah, that's good advice. That's good to
yeah,
know. Well, it was so nice to talk to you, and
it
thank
was
you so
very nice to
much
talk to you. I'm
for
sorry I didn't get to
making
meet
time.
Gilbert, but
Oh, I know. This child,
yeah,
he's like, well,
you know
I'm
what?
just
They
gonna
nap when
pass
they
out.
nap, and that's that. So
They
yeah,
do. It's like he runs the show for sure.
yeah, yeah,
Yeah, well, it's great to talk to you. And
yeah.
yeah, hopefully one day we will make it out there and we'll get to meet you in person. And we'll
You're in
get to
great
do
hands
this
where you
again.
are. So
Yeah,
yeah, but we'd love to
we
meet
are.
you.
Yes, yes. It would be fun. All right. Well, thank you so much. And yeah, this will probably be up in a couple of days. So thank you so
Good.
much for
Take
talking.
care. Have a nice weekend.
You too.
Bye bye.
Okay, bye.